An operon is a functioning unit of DNA containing a cluster of genes under the control of a single promoter. It is comprised of an operator, a promoter, and one or more structural genes that is transcribed into mRNA. A lac operon is an operon that is required for the transport and metabolism of lactose and E. coli and numerous other types of bacteria known as genetic regulatory mechanisms. Lac comes from the beginning of the inducer molecule of the operon. This operon allows lactose to be digested when glucose is not available through the activity of beta-galactosidase, which LAC-Z gene codes for. It cleaves lactose into galactose and glucose. Next is the LAC-Y gene that codes for the lactose permease, which is a cytoplasmic membrane protein that transports lactose into the cell. Lastly, there is the LAC-A gene that codes for beta-galactoside transacetylase. However, only the LAC-Z and the LAC-Y is needed for lactose catabolism, which makes LAC-A not as important to understanding the whole process. Now, there's also two other regulatory sequences consisting in the lac operon: the promoter, which can bind with RNA polymerase so transcription can occur. Next, that sequence is the operator, and upstream of that is two other regulatory genes of the DNA, called the repressor protein and the associated repressor promoter. Now, when glucose is ready for use of the cell, the repressor protein is considered expressed. Then this regulatory protein binds to the lac operator. This interferes and represses with the binding of RNA polymerase, which wants to bind the lac promoter. Because of this, it prevents transcription of mRNA. This will then change when glucose is not readily available and another source of energy is available in the form of lactose. Lactose enters the bacterial cell, and a metabolite of lactose called allolactose binds to the repressor, which will alter the shape of the repressor, causing it to fall off the operator. Now that the repressor has released itself from the operator, RNA polymerase is free to transcribe all the three genes, LAC-Z, LAC-Y, and LAC-A. Now this will lead to higher levels of coded protein, beta-galactoside, and permease. Permease will allow for more and more lactose to enter the cell, which can be broken down by beta-galactoside to make lactose and glucose, which will then be used for the cell's metabolism. In the case of both lactose and glucose being present, however, the transporter glucose will block the transport of the inducer molecule, lactose, also known as the inducer exclusion. The transferred glucose will form a protein that will bind to the lactose permease to prevent it from letting in lactose. Now, with the decrease of lactose, which will leads to decreased repressor protein binding, it will then latch back onto the operator and prevent RNA polymerase, like already stated. Now we will talk about the terp operon. This operon consists of a promoter and an operator where the repressor will bind, such as shown in the lac operon. Downstream of the DNA are 5 different structural genes that code for enzymes to therefore make tryptophan, terp E, terp D, terp C, terp B, terp A. Now let's pretend the cell is experiencing low tryptophan. It needs tryptophan as a building block to build proteins. The RNA will bind to the promoter and begin to transcribe the genes into mRNA, which will then be translated into protein. These will then create more tryptophan through the process of tryptophan biosynthesis. In the next case, the cell experiences more tryptophan. At this point, the cell doesn't want to make any more when it is no longer needed. Tryptophan can also act as a co-repressor for the repressor molecule for a repressor enzyme that has the ability to attach to the operator and stop RNA polymerase from transcription. The repressor then becomes activated, and the whole process starts again. Now, these are the ways a cell can regulate their genes, which is essential for the cells to survive.